In this video, we'll be seeing about the pathogenesis of tuberculosis. Before we dive into pathogenesis of tuberculosis, let's say in general what tuberculosis is. So tuberculosis is a chronic pulmonary pertaining to lungs, and systemic disease, systemic affecting the whole body, caused most often by Mycobacterium tuberculosis, and it is the leading cause of death, infectious cause of death worldwide. So when I say it is most often caused by Mycobacterium tuberculosis, you can also have other strains causing it, like Mycobacterium bovis, which results in oropharyngeal and intestinal tuberculosis. It is rare in countries where milk is pasteurized, but still it is seen where there are tuberculous dairy cows and unpasteurized milk. Okay, fine. So coming to pathogenesis of tuberculosis, now in this mechanism, we're going to see about how the infection is manifested and what results it brings about in a previously unexposed and immunocompetent individual. In a previously unexposed and immunocompetent individual, meaning it is occurring for the first time. So this is primarily based on the development of antimicrobial T cell mediated immunity. So now this T cell mediated immunity, as you would have seen, as you would have learned in microbiology, it comes under your adaptive immunity. So yes, so we are going to see in an unexposed immunocompetent person, which results in the development of an antimicrobial T cell mediated immunity. Now, before we start, let me just introduce, this is Mycobacterium tuberculosis. This is our alveolar macrophage. And turning on to the next, this is the alveolar macrophage with a major histocompatibility complex. I will be explaining what each of these mean in simple terms as we go through. This green is the antigen. We have our T cell here, which differentiates into the Th1 cell, which and then we have, this is our alveolar macrophage. So just take a look at the color codings, like yellow, and purple over here, purple over here. Okay, now starting off. Okay, before we go, what is alveolar macrophage? What is macrophage? Macrophages are basically, we know about monocytes. Monocytes are WBCs, right? So monocytes are found in blood, but when the same monocyte migrate to tissues, they are called macrophages. And the main activity of these macrophages is phagocytosis and antigen-presenting cells. Meaning, in simple terms, you can just say that they go on, pick the bad guy, which is the antigen or the foreign particle in your body, and they try to bring about immune responses to destroy them by presenting it to antigen-presenting cells. Who do they present it to? They present it to your T cells. Okay, starting off, we have this Mycobacterium tuberculosis, right? So this Mycobacterium tuberculosis, they enter into the macrophage, your alveolar macrophage, via the receptors like receptor 4, sorry for that, via the receptors like mannose binding lectin and type 3 complement receptors. These are just fancy names. Phagocytosis, yes, phagocytosis, we know that macrophages are principal cells involved in phagocytosis. How do they bring these about? By recognition, engulfment, fusion, killing, or degradation. These are the steps involved in phagocytosis, which is altogether a different topic. So if it was normal, say if it was not Mycobacterium tuberculosis, if it was just another normal bacteria, it would have formed a phagolysosome inside the macrophage, which is followed by destruction. So it is killed, and we do not manifest any kind of infection. But in case of Mycobacterium tuberculosis, it inhibits the maturation. This Mycobacterium tuberculosis, it inhibits the maturation of phagolysosome. So lysosomes are nothing but they bring about the microbicidal mechanisms, meaning they kill the bacteria. So when this Mycobacterium tuberculosis inhibits the formation of phagolysosome, it allows the bacteria to multiply unchecked. So it can keep on multiplying. There's nothing to do with it. So how does this happen? This happens when the bacterium blocks the phagolysosome formation by recruiting a host protein, which is called coronin. So this is called coronin, and this coronin is a host cell protein. Now this coronin, it activates phosphatase calcineurin, and this phosphatase calcineurin inhibits the say the fusion of phagosome and lysosome, and hence it does not allow the phagolysosome to be formed. So that is about, the Mycobacterium tuberculosis enters via various receptors. If it was normal, it would have undergone, a phagolysosome would have formed and it would have been destroyed. But since this is not happening, we're having the recruitment of a host cell protein onto the surface of the phagosome, coronin. It results in the activation of phosphatase calcineurin, which inhibits phagolysosome formation. So ultimately, what happens? There will be unchecked replication of these bacteria within the phagosome, which itself is contained within the macrophage. Okay, so what does this manifest? We can see bacteremia with seeding at multiple sites, as you can see in this gross picture here. You have these tiny, tiny white particles which are not normally seen in the cut surface of a normal lung. So this is how it manifests. Bacteremia with seeding of multiple sites. So in this stage is often latent, which means that this happens in less than 3 weeks after infection. So mostly in this stage, the individual is asymptomatic, or he or she can have mild flu-like illness. So we saw what happens before 3 weeks. Now what happens after approximately 3 weeks? So at the period from slide 1 to slide 2, as we switch over, certain innate immunity mechanisms kick in. Say we have read about these pathogen-associated molecular patterns. Pathogen-associated molecular patterns. Now, these pathogen-associated molecular patterns, they bring about the interactions which initiate and enhance the innate and adaptive immunity response to Mycobacterium tuberculosis. Now, after about 3 weeks, when your antigen it passes through the lymph node, is when all these reactions happen. So now let's see what happens. Now, this is your alveolar macrophage, right? This is an antigen-presenting cell too. This has the host cell surface molecule called the major histocompatibility complex. Now, this major histocompatibility complex, its job is to bind to the pathogen and to aid in its recognition by the T cell. So, when we read about B cell, we got to know that in B cells, they can just go about and directly attack the antigen. But T cells cannot do that. They need a special person, the macrophage or any other antigen presenting cell, to take up the antigen and process it and then present it for recognition by the T cell. That is the whole mechanism involving your antigen presenting cell, which is your macrophage, the major histocompatibility complex, and the T cell. Now, the major histocompatibility complex, why it is named histocompatibility is because it is usually involved in, say, a graft rejection. That's a totally different topic. We'll come to that later. So what happens in this reaction is that so the response is initiated by mycobacterial antigens when they drain into the lymph nodes, right? So this results in the production of interleukin 12 and interleukin 18. Now, interleukin 12, when you look at what is the function of it, it results in Th1 cell induction, meaning after this whole recognition process happening here, the interleukin 12 produced by the alveolar macrophage results in the differentiation of this T cell right here to Th1 cell. Now, this Th1 cell, it is very specific to bring about the processes of cell-mediated immunity. Now, this is over. You know how the Th1 cell comes about. Now, what does this Th1 cell do? This Th1 cell, it is mainly concerned with macrophage activation. So it is mainly concerned with macrophage activation, and also it results in the killing of bacteria. How does it do that? So the Th1 cells, they produce interferon gamma. Th1 cells produce interferon gamma. Now this interferon gamma, it is a critical mediator which activates the macrophages, and it enables them to contain the Mycobacterium tuberculosis infection. Now, the whole point of why a caseous necrosis is formed, or why there is a Th1 cell coming in, why there's T cell mediated immunity, is because when your body cannot eradicate a pathogen, it switches over to mechanisms to contain it. And thus, it can also result in tissue damage. We'll see how that happens. So coming back again, you have this alveolar macrophage producing interleukin 12. Interleukin 12 results in differentiation of T cell to Th1 cells. This Th1 cells produces interferon gamma. Interferon gamma, when it acts upon the alveolar macrophages, results in their activation and killing of macrophages. Now, by what mechanisms are these macrophages killed? First mechanism. What did we see in the previous slide? That they inhibited the maturation of phagolysosome. So what happens here? The maturation of phagolysosome kicks in. So the lysosomal enzymes can take over and kill the bacteria. The other mechanisms are the interferon gamma. It stimulates the production of nitric oxide synthase. This nitric oxide synthase eventually results in production of reactive nitrogen intermediates, which are very important for killing of bacteria. And also, with all these going on, you have a lethal acidic environment. Added to it is that interferon gamma, it mobilizes antimicrobial peptides, say defensins against the bacteria. So finally, all these mechanisms lead to autophagy, which destroys the damaged organelles and the intracellular bacteria, which is your Mycobacterium tuberculosis. Remember, all this while your Mycobacterium tuberculosis was within the macrophage, intracellular. That's how it came about. Now, what happens after this? We have your Th1 cells, which release interferon gamma, and which results in activation of macrophages and killing of the Mycobacterium tuberculosis. What happens next? We are done with killing of macrophages. This part is done. So what does this activated macrophages mean? Well, activated macrophages mean that under the influence of interferon gamma, these macrophages, they change their shape, they change their form, or they differentiate into these epithelioid histiocytes. You can see these cells grouping around the area of necrosis, which we'll come to this later. Okay, now caseous necrosis, it is nothing but it is caused by, these activated macrophages, they secrete your tumor necrosis factor and chemokines, which results in the formation of caseous necrosis. Caseous meaning it looks cheesy in gross appearance. Now, macrophages, they differentiate into epithelioid histiocytes, and they are surrounded by these T lymphocytes. Now, what structure do we have here? This is the typical structure of a granuloma. Granuloma contains activated macrophages. Granuloma contains activated macrophages in the form of epithelioid histiocytes, abundant cytoplasm, and a collar of T lymphocytes, depending on whether the granuloma has taken, like if it is old, if it is chronic, if this condition is chronic, you can see some fibrous tissues surrounding it. So this is how your granuloma forms. See, this is the cut surface of a normal lung. But here you can see something which is cheesy, friable in appearance, which is your caseous necrosis. This caseous necrosis. Now, also important to note that when one granuloma, called tubercle in the case of tuberculosis, when one tubercle is formed, you will not get this caseous necrosis. This results only when there are multiple granulomas, like more than one tubercles, results in the appearance of a caseous necrosis. So that's how this is formed. Going through quickly, we have this Mycobacterium tuberculosis here, where this Mycobacterium tuberculosis enters via the receptor into the alveolar macrophage. It inhibits the maturation of fusion of phagolysosome. So hence it replicates freely inside. At a point approximately after 3 weeks, it is sensed by the pathogen-associated molecular patterns of the innate immunity, which kicks in the following responses. presented by the major histocompatibility complex to the T cell. Your T cell mediated immunity starts. This T cell differentiates under the influence of interleukin 12, interleukin 18 into Th1 cell. Th1 cell produces interferon gamma, which results in activation of macrophages and killing of microbes by various mechanisms such as maturation of phagolysosome, lethal acidic environment, reactive nitrogen intermediates, defensins. All this leads to autophagy and killing of the microbe. And here you have the activated macrophage, which changes its form to epithelioid histiocytes. The granuloma, which is surrounded by a collar of T lymphocytes, epithelioid histiocytes, in the center of caseous necrosis. Caseous necrosis is a result of secretion of tumor necrosis factor and chemokines. And here we can see the gross appearance of what a caseous necrosis looks like. We can see some cavitation here, so it might be a picture of secondary tuberculosis. You have two types of tuberculosis, primary tuberculosis and secondary tuberculosis. And this is a picture of which is the non-comic version of it. So you can see here the alveolar macrophage. It is the same thing. The same Mycobacterium alveolar macrophage replication, the same class 2 MHC antigen T cell receptor, T cell differentiates to Th1. Tuberculin test is based on it, based on this, and you have mycobacterial killing and caseous necrosis. So that's how the infection with Mycobacterium tuberculosis results in formation of a granuloma. In fact, tuberculosis is the prototype of granulomatous inflammation. So when you suspect, when you see a granuloma, you have to first exclude tuberculosis. That's the primary cause of it. Also, another fun fact, tumor necrosis factor, it's often neglected. Say when in rheumatoid arthritis, when the patients are treated with tumor necrosis factor antagonist, TNF antagonist, they have an increased risk of tuberculosis reactivation. So that's there. Hope you understood. Feel free to share any information, or if you want to see or if you want to know any topic in the comments below, like and subscribe. Thank you.